right, so we are here to talk a little bit about what I'm going to call foundations of gene expression. So it's just some kind of background information that you need to know to understand gene expression. And the first is I can define gene expression. So gene expression is, you're going to hear it sometimes called protein synthesis. That's not 100% a synonym, and I'll talk about why in a second. Or you may hear it called transcription translation. Gene expression will always involve transcription. It may or may not involve translation. So gene expression is simply converting the code or decoding the code in DNA into a molecule that the cell can use. Okay, so we're taking those A's, T's, C's, and G's in DNA, and we're doing transcription to make an RNA, and either that RNA has a job to do and it goes off and does that job, or that RNA is mRNA and it goes to the ribosome for translation or protein synthesis to make a protein that has a job to do. So we used to say that gene expression and protein synthesis were the same thing, but we now know that not all RNAs become protein. Some of them have jobs to do, and so we can't really say they're the same thing because you're not always making proteins with gene expression, but that is one direction you could go. So that's what gene expression is. For a long time, originally, and I talked about this in another video a little bit ago, that they thought proteins ruled the world, and proteins do, but proteins are coded for by DNA. So proteins rule the world, but they rule the world because they do all the chemical reactions, but DNA is responsible for making those proteins. So while proteins are super duper important, if there were no DNA, there would be no proteins. So by default, DNA is super duper important as well. But we didn't know a lot about DNA at first. We knew a lot more about proteins before we knew much about DNA. And so originally, they studied the genetics, kind of how genetics worked, gene expression, and they've thought that every gene, every segment of DNA that starts with a promoter region and ends with a terminator region, that chunk of DNA coded for an enzyme, and that's what your DNA did. It coded for enzymes, and they said that's great. Then later they realized, well, not all proteins are enzymes, because at that point it was kind of like, well, they code for proteins, but all proteins are enzymes. Then they said, no, not all proteins are enzymes. There are proteins out there that do jobs that are not enzymatic in nature. They're not catalytic in nature. And so the new kind of idea, the new statement was, well, now we think one gene, or we're going to say one gene becomes one protein or one polypeptide, which is another word for protein. Because, DNA becomes RNA, RNA becomes protein. Well, then more recently, it's not that they discovered new information, but they realized the wording wasn't perfect there. So they've tweaked that, and I say recently, not like in the last year. I mean, it's been a while, but they've said, no, now we know that every gene in DNA codes for RNA, but not all RNAs become protein. So we don't need to say one gene makes one protein, because that's not true. We have DNA genes that code for tRNA and rRNA and a bunch of other RNAs. And so now we know that one gene codes for one RNA. All right, so that's the new, most accurate, most up-to-date model. The next I can statement is I can explain central dogma. Well, central dogma, a dogma is just a statement of belief. And a central dogma is an important, like central to the understanding of a concept statement of belief. And so central dogma is the central driving idea behind genetics, and that is that DNA codes for RNA that codes for protein. That's the flow of genetic information. Now we know sometimes this takes a different path, and so sometimes it's just DNA to RNA, and that RNA goes and does a job. So there's kind of two pathways you could look here. So DNA to RNA, stop, or DNA to RNA to protein. I can compare and contrast DNA and RNA. So let's look at a little table that I drew, which, gracious, my lines aren't straight. And let's talk about some ways that DNA and RNA are similar and some ways that DNA and RNA are different. Well, first of all, they are both nucleic acids. They both are made of nucleotides. They both have A, G, and C, adenine, guanine, and cytosine. They both have a 5-carbon sugar. They both have phosphate groups. They both are involved in transcription. They both can be found in the nucleus. Okay, so lots and lots of similarities. And then how are they different? Well, DNA is double-stranded. and RNA is single-stranded. DNA has thymine, where RNA has uracil. DNA's sugar is deoxyribose, where RNA's is ribose. That just means that DNA has one less oxygen, so deoxy. What else? DNA stays in the nucleus. RNA can leave the nucleus. RNA is involved in translation. That's that process of taking RNA to protein. Not all RNAs, but some of rRNA, tRNA, and mRNA are all involved in that process, whereas DNA is involved in replication. There was something else that just went through my head. Oh, there's really only one type of DNA, and there are many types of RNA. I mean, the big three that we talk about are RNA, mRNA, and tRNA, but you're going to learn about even, or you've already learned hopefully about even more. So many types of RNA. So that's just a few. There might be others you thought of that you want to add to your list, and that's perfectly fine. But those are just a few. I hope that helps. Oh, I'm not done. Just kidding. Just kidding. I can discuss the many roles of RNA. So let's look at this. I do hope that helps, but I'm not done. So in eukaryotes, when we go through transcription, we make something called pre-mRNA. Now, you don't talk about this in Bio 1, you just call it mRNA because it's Bio 1 and it doesn't really matter. But now that we're going a little deeper in AP, what comes off of the DNA is actually not ready to go to the ribosome. It has to be edited. And we'll talk about that in another video. So once it goes through that editing process, then it becomes mRNA that can leave the nucleus and go to the ribosome for translation. There is tRNA. tRNA I like to think of as a taxicab, or an Uber. It stands for transfer RNA. M is for messenger RNA because it's taking the message of DNA out to the ribosome. tRNA is transfer RNA because it's transferring amino acids from the cytoplasm to the ribosome. So from one place in the cytoplasm to the ribosome. And so it binds to a specific amino acid. It has an anticodon on it that matches the codon on the mRNA. And so if the mRNA is AAA, the tRNA is going to say UUU, and there's a particular amino acid that goes on that specific tRNA. And so when it comes in and those two bind, then it drops off that amino acid. And so that's how we know that we're going to get the right amino acid. But we'll talk about that more next week. rRNA is made by DNA, but it makes up the ribosome. So rRNA actually goes into the nucleolus. And there are some proteins that are made that go into the nucleolus, and then boom, 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 out pops ribosomes. And then those ribosomes go out to the cytoplasm to do their job. There's small nuclear RNA that is part of a spliceosome, which is part of that splicing reaction that's RNA and proteins work together to cut that mRNA. And so there's some RNA in there that has some structural roles, meaning that it holds the structure in place. It also has some catalytic roles, some chemical reaction roles. SRP RNA is basically like a zip code kind of. It tells proteins where to go. So, hey, you need to go to the mitochondria. Hey, you need to go to the Golgi apparatus. Hey, you need to go to the to be bound to the rough ER or whatever. So it tells that where to go. There's interference RNA, which we call RNAi. It's going to regulate things because it's interfering. And then there's ribozymes, which is just the term we use for any RNA molecule that can function as an enzyme. Remember, enzymes are things that decrease activation energy to help a chemical reaction occur easier. And so any RNA that does that is called a ribozyme. And then the last is I can define a codon. A codon is 3 bases on an mRNA molecule, and it is read. mRNA is read in 3s, and so we call that the reading frame. So it reads these 3, and then it shifts over and reads these 3. There's no overlap. So for example, if this is your, whoops, there's not going to be a T. If this is your mRNA, it would read those 3, and it would read those 3, and it would read those 3. Each one of those would code for an amino acid. So this would be 9 bases, 3 codons would bring in 3 amino acids. And that's what a codon is. So now I'm done. I hope that helps. If you have any questions, let me know.